Good morning, students. Welcome to week 3. Today we have lecture 6, where we focus on the internal structure of the cell, particularly the cytoskeleton, and related topics. I want to first put this lecture in context, as we always try to do. We're in our first unit. We are defining the cell and its study. We are now in our third lecture on structure of the cell. The first of these lectures focused on dynamics at the surface of a cell, focusing on the structure of membranes and the proteins embedded within, and the movement of material across those membranes. The second lecture within the structure of the cell focused on the movement of material through the cell, the movement of material out through the secretory pathway, and then bringing material back into the endosomal lysosomal pathway. The third of these lectures focuses on internal structure. The bulk of the lecture will be focused on the cytoskeletal components. The reading is out of chapter 17 for Alberts et al. We're going to break this lecture into 2 parts. Part 1 is going to focus on these 2 bullet points, where I will first introduce the components of the cytoskeleton. Then we're going to focus on actin, or the microfilaments, take a look at how they get polymerized, how interacting proteins help to stabilize or break down the filament structure, the role of actin, and then the involvement of actin in muscle contraction. We're going to do one specific example related to actin. That will close out part 1, and then I'll be back after a short break to record part 2, where I'll focus on another component of the cytoskeleton, this time the microtubules. Again, take a look at how they're polymerized and organized, how material moves along the microtubules. And then I'll link this lecture to our lab, and we'll focus on a newly discovered role for our friend from lab, APP, in mediating kinesin movement along microtubules. This is an important part of the secretory pathway and the movement of material out of the cell. So we'll try to link what we're doing in lecture today to lab. Okay, first, my introduction to the cytoskeleton. We're going to focus on them sort of systematically here, beginning with the smallest of the cytoskeletal components, the actin filaments, and then kind of working our way down to the largest, the microtubules. Beginning with actin, about 7 nanometers in diameter. Actin is a filament, meaning it's a polymer with highly stabilized structure. Here is an electron micrograph of what that filament looks like. It's like a string. It almost looks like pearls on a necklace when we look at it in the electron micrograph. And here's a cartoon rendering of the actin filament. You can see it has some spiral nature to it. So there is actually a spiral-like structure here. And all of the filament is made up of monomers of actin. Actin is a protein product. It comes in different flavors in humans, for example. There are half a dozen or more different isoforms of actin. In yeast, the simplest eukaryote, there's only one. But whether it's a mix of these monomers or just a homogeneous collection of these monomers, they arrange themselves in a very particular way, and then they localize to the periphery of the cell. Take a look here. We're looking at an epithelium. You may recall from one of my previous lectures we talked about epithelial cell sheets in the gut and the role of the basement membrane down here, that specialized ECM. Well, here's another look at the epithelium. And wherever you see the pink, you can see the pink color here, including up here in the microvilli of these epithelial cells. That's where we would find actin. There's a high peripheral concentration of actin in the cell. It tends to localize to the periphery. And it localizes just under the inner leaflet of the lipid bilayer, so right at the cell surface. One of the most important things that actin is doing there up at the cell surface is anchoring membrane proteins and helping to keep them from diffusing in the lipid bilayer. Other things that actin will do, we'll see a role in actin in muscle contraction. That's one of its most important roles in myocytes. Myocytes are muscle cells. When they contract, it has to do with sliding filaments of actin. We'll take a look at that a little bit later. Next up, if we're moving from small to big, are the intermediate filaments. Intermediate filaments are also polymers. You can see what they look like in the electron micrograph here. The intermediate filaments tend to be arranged orthogonally, or at 90-degree angles. And these filaments, unlike actin, are quite heterogeneous. There are 50 different types of unique protein products that can serve as monomers to build the intermediate filament polymer. And the intermediate filaments are very, very strong. They're in high concentration in cells that are prone to mechanical stress. So cells that have to change their shape often, for example. They're very important in epithelia at forming what we call desmosomes. Desmosomes are here, and they form these really tight connections between the epithelial cells and help to prevent material from diffusing down towards the basement membrane. We won't talk very much about intermediate filaments in today's lecture. I just want you to be familiar with them as a class of cytoskeleton and understand that they're a sort of heterogeneous type of polymer. The third cytoskeletal component, and the one that we'll focus on in part 2 of today's lecture, are the microtubules. And these are the largest. They're at about 25 nanometers in diameter. Here's a look at the electron micrograph. They tend to be arranged kind of linearly and like train tracks is the way I like to think about them, to allow for material to move through the cell. And the types of materials that move along microtubules, they do so on motor proteins. Motor proteins sort of bind to the microtubules and then connect to vesicles. And those vesicles could be loaded with protein cargo moving through the secretory pathway, or protein cargo that's been endocytosed and now making its way to an endosome. Or we could have organelles carried by these motor proteins. And the motor proteins use ATP hydrolysis to sort of move in one of two directions along the train tracks. The other place where you might have heard, or a place where you might have heard of microtubules, is their role in mitosis. They're very important in the separation of the sister chromatids in anaphase. So sort of moving them from the metaphase plate towards the two daughters. Today we're going to focus on the role of microtubules in transport of material through the cell. Now that we've seen our introduction to the components of the cytoskeleton, we're going to focus on actin for the rest of part 1. First, I'll give you a little bit of an introduction to the way they assemble, the actin filaments assemble. Then we'll look at binding proteins and then the role in muscle contraction. So first thing to note, actin is quite an important protein, and we can get a sense of that by knowing that about 5 to 10% of all protein synthesized by the cell is actin. And actin begins as this sort of folded globular structure here that we call G-actin, globular actin. Now there's polarity to this molecule, and the artist has tried to draw it. You see it looks like almost like a little pie, and on one end of it there's a little slice of the pie that's colored black. That's trying to indicate the polarity of the molecule. And the little part where the black piece of pie is the minus end of the molecule, and the other side is the plus end of the molecule, and that's just an arbitrary designation that we use to draw your attention to the fact that the molecule is polar. Now, globular actin doesn't do much of anything. We need to assemble it into a filament, into a polymer, for it to have any structure and for it to be able to perform its role in anchoring proteins and all those other things. So there's a nucleation step in the assembly of the actin filament where a particular protein that I'll introduce later forms a collection of globular actin monomers. And we call that collection a nucleus. Now don't confuse this with the organelle nucleus. It's kind of an unfortunate use of it's like a homonym, I guess. We're using the similar word to mean something completely different. But anyway, we get a nucleus or a collection of globular actin monomers, and now from that, we can very quickly begin adding monomers to the plus or the minus end. And that begins the elongation phase. Now, what you'll notice here in the cartoon is that monomers are added a little bit quicker to the plus end than they are to the minus end. Okay? And at some point, we get to a steady state where the filament is of fixed length, and the addition of monomers to the plus end is equally offset by the leaving of monomers from the minus end. That steady state is also called the treadmilling state, and it's analogous to the treadmill that we would run on in the gym. The length of the treadmill doesn't really change, right? It stays the same as we run on it. A couple of graphs here to focus on, and this is showing us the role of nuclei in helping to speed up the rate of filament formation. So we're looking at the mass of a filament over time, and we see a real nice shift to the left here indicating an increase in the rate of nucleation when nuclei are present. So when we have this nucleation phase, we dramatically speed up the rate of filament formation. And obviously the filament would still form even without nuclei. You can see it just takes a little bit longer to get to the plateau phase of the graph. And that same concept is, or a similar aspect of that is illustrated here. We're looking at the mass of the filament over time again. And once we reach the critical concentration of nuclei, we switch from this sort of linear monomer phase to a linear filament phase very quickly. And so nucleation does help to speed up the reaction, and it helps to keep the cells in that dynamic phase of very quick actin polymerization. Cells need to be able to synthesize and make filaments of actin very quickly, so having nuclei allows that. Similarly, sometimes cells need to dissociate the filament very quickly, and there are ways to do that I'll show you on a subsequent slide. Here we're looking at a closer, sort of zoomed in look at the actin filament, and we're kind of trying to model the treadmilling phase here. We can see this filament is of fixed length. And what I want to draw your attention to is here's the plus end of the filament, here's the minus end of the filament. Take a look at the monomers of actin that are here at the plus end. They are bound to ATP. And the artist has shown us the ATP and the 3-phosphate groups there, and they've drawn it in red. Whereas at the minus end, all the monomers have ADP, and that's kind of interesting. How do we get from ATP to ADP? Well, it turns out that actin has an intrinsic ability to hydrolyze its bound ATP. So actin, in a way, is an ATPase. An ATPase is an enzyme that can hydrolyze ATP, and it doesn't do it, it doesn't require anything other than itself to do it. It's kind of interesting. I think of this as sort of like a timer mechanism. So the ATP-bound monomer, the ATP is put on, and then we start a timer. And the ATP will stay in this ATP state for a period of time. Once the timer goes off, the hydrolysis activity kicks on, and now we hydrolyze it to ADP. And the reason for that is disassembly happens much faster when the monomers are in the ADP-bound state, and that's because the filament is a little less stable in the ADP-bound state. So the dynamic nature of actin is facilitated by that ATP hydrolysis. If we always had ATP-bound monomers, it wouldn't be able to be broken down very easily. So it's almost like a way of carefully regulating the dynamics via hydrolysis. At some point, we need to regenerate the ATP-bound monomers from ADP ADP bound monomers, and that happens through nucleotide exchange. So we kick off the ADP, so the ADP leaves the complex, and then we bring in the ATP. So it's true nucleotide exchange. It's not just let's phosphorylate that ADP to make ATP. It doesn't work that way. It's actually nucleotide exchange where this is thrown out and this is put in. So now we're looking at actin binding proteins, and these are other proteins that can interact with actin either in the monomer or the filament stage, and they help to affect the dynamics. So let's look first over here at the role of cofilin in breaking down an actin filament. So I'm going to move the slide over a little bit so I can see this better. There we go. Okay, so we're looking here at a filament of actin. Here's the plus end, and the plus end, remember, has the ATP-bound, more stable monomers, whereas the minus end is full of ADP-bound monomers, those less stable versions. Cofilin is an actin binding protein that helps to speed up the rate of filament dissociation. And it does it through a couple of different strategies. One strategy is simply to have a high affinity for the ADP-bound monomer, and the ADP-bound monomers are more attracted to cofilin than they are to each other. So if there's a good amount of cofilin in the cell at any point in time, it's going to be able to sequester or trap or grab those ADP-bound monomers from the actin filament. The second strategy that cofilin utilizes is it has a way of breaking the weak force attractions that hold the ADP-bound monomers together. And what this what this does, it's not actually cutting, and that's why I put kind of cut in quotes here. Metaphorically, it's cutting, but it's not literally cutting. It's not hydrolysis. It's just simply breaking the weak force attraction. So it's similar to cutting in the sense that it forms multiple minus ends, and that will make dissociation happen much more rapidly. So cofilin, an important actin binding protein that helps to affect the disassembly phase. Why would a filament of actin need to be disassembled? You know, why? Let's ask that question and answer it. It's worth asking. It may be difficult to imagine an example of where that might be helpful, but I want you to think about it like this. What if you're an immune system cell that's traveling in the blood, and there's been some tissue damage in the periphery? You've, stubbed your toe or have a splinter in your finger or something, and there's tissue inflammation, and there are signals that are trying to attract those immune system cells to that tissue to help repair the damage. How in the world can an immune system cell make it from the lumen of a blood vessel where the blood is flowing into the connective tissue and then into the skin? Well, it's going to need to very quickly change its shape and squeeze through the cells that are lining the blood vessel. So the cell will pause and it will quickly disassemble its actin filaments to change its shape, to flatten up, go from a sphere to like a disc. And then it's able to squeeze through the cells that form the blood vessel wall. So that's one example of where a cell might want to very quickly disassemble its actin cytoskeleton and then change shape, and then when it gets on the other side, reassemble back into its normal shape. Okay, let's look at another actin binding protein. This one's called profilin. Profilin promotes filament formation. That's how it gets its name, profilin. And profilin is essentially a nucleotide exchange factor. So profilin stimulates the removal of ADP from an actin monomer, and in turn, and consequently then stimulates the introduction of ATP to that same monomer. So on the previous slide, when I was talking about regenerating the ATP-bound globular actin, Profilin is the protein that's promoting that. So now we see profilin bound to the ATP version of globular actin. And then what profilin does is it delivers it to the nucleating protein. The nucleating protein, the one that takes the 3 or 4 or 5 globular actins and forms a nucleus with them, that protein is known as ARP23. That's the name of that. It's a weird name, but that is the name of the protein. And once the nucleus is formed, the ARP23 moves away, and now we're ready to add ATP-bound monomers, preferentially to the plus end of the filament. And you can see that profilin is helping to aid in that process as well. Other actin-binding proteins help to stabilize the filament in the ATP-bound state. And those proteins are called capping proteins. And those capping proteins, they're shown kind of as a blue arrow here. They associate with the plus end of the filament, and what they do is they tone down the ATPase activity of actin. And if they help to tone it down, meaning that it doesn't work as well, that means that the timer is going to be extended. So, we're in our countdown for ATP hydrolysis, we cap, and then we extend the timer. And now it takes longer for that ATP hydrolysis reaction to kick on or turn on. And the result of that, of course, is we have a more stable filament for longer. So when would we want that sort of dynamic to occur? When would we want to cap our actin and keep it in the stable phase? Well, we might want to do that in a myocyte. A myocyte is a muscle cell, and the muscle cells kind of line up in series in a muscle fibril or myofibril. And sliding of cytoskeletal components helps the myocytes to sort of shorten from both ends, and that gives us our contracted muscle. And capped actin filaments are the predominant type that we see in myocytes, and that helps to keep them stable and helps to help them function in the unique environment of the muscle cell. And I'll give you a little bit more of a sense of that in the next couple of slides. All right, before we get to the role of actin in muscle contraction, let's take a look at what actin's doing in all cells up at the cell surface. And what actin is helping to do is form the so-called cell cortex. And the role that it's playing is kind of shown in these two cartoons. Let's focus on this one first. Here's the cytosol. Here's the extracellular space. Here we see the lipid bilayer. This is the outer leaflet. This is the inner leaflet. And we see these integral membrane proteins, these purple guys here and this one here. These proteins contain alpha helical transmembrane domains. The blue protein has 2 of those TM domains. The purple one has more than I can probably see or count here, but many more. And these are, remember, helping to anchor the proteins in the membrane. And on this side, the extracellular space, we have the extracellular portion of those integral membrane proteins. And we can see that they've been glycosylated. See all these sugars that are added here. That happens in the ER and the Golgi of the secretory pathway as the protein's moving its way up to the cell surface. Those sugar groups get added. We don't typically see glycosylated proteins facing the inside of the cell. They typically only face, glycosylation sites rather, are typically only found on the outside of a protein. What's not shown in this cartoon on the left is the extracellular matrix, but I'll kind of draw it here. Extracellular matrix is there, and these integral membrane proteins, some of them anyway, are also interacting with the ECM. We talked about how that helps to provide a little extra stability for animal cells in a previous lecture. Well, here's what actin is doing on the inside of the cell. Notice that a filament of actin is shown here. I've just kind of colored it in. And there are some binding proteins, the red ones, interacting with that actin. And they're helping to link the actin to another protein called spectrin, which runs kind of longitudinally along the inside of the cell. And every once in a while, we don't see another one on this view, but there's another actin filament here that's helping to anchor in all these different cell cortex components like spectrin. And what we're doing here is forming a really strong matrix at the surface that's helping to keep these integral membrane proteins in place. If we didn't have them linked up to the cytoskeleton, those proteins would be able to diffuse freely within the fluid mosaic of the lipid bilayer, and they might move away from each other. That could be problematic if the purple proteins and the blue proteins are part of some signal transduction pathway, for example. We want to keep components of a pathway together. We don't want them randomly diffusing through the cell. So actin can help to anchor those proteins. Here's another look on the right side here. Now we've kind of reversed the diagram. Now this is the cytosol, this is the extracellular space. We see the extracellular matrix that's formed and how it's interacting with this protein, integral membrane protein here, at the surface of the cell. Now on the inside, a different component, like spectrin in the previous example, but this one's called dystrophin, is helping to link this integral membrane protein to the actin filament. And again, that's going to help to stabilize the proteins in the surface of the cell. Dystrophin is an important protein from a clinical perspective. It's one that's either mutated, loss of function, or missing, sometimes deletion, in muscular dystrophy. And that's a disease of the muscles, the myocytes, that helps to prevent normal movement. So that gives us a sense of how important proteins like dystrophin and their interactions with actin are in normal cellular function. Okay, as promised, we'll now take a look at actin and the role that it plays in muscle contraction. And let's first introduce a little bit about the anatomy of the muscle. So we're looking here at a cartoon schematic of the biceps. If we sort of take what's inside and expand it, we see that there are these bundles of muscle fibers. We call them muscle fibers. And each muscle fiber contains about 1,000 of these individual myofibrils. Myo, that prefix means muscle, and fibril, of course, means like fibril, filament, fiber. You can think of it, the root there is similar in all of those words. If we look at the myofibril, an individual one, we see that it's formed by the fusion of all these different cells together. And you can see that it looks striped. I'm going to try to color this in, but here's like a darker part, followed by a lighter part, followed by a darker part, lighter, darker, lighter, etc. That's how we get the word striation. Striation is a fancy way of saying stripe. So striated muscle, or skeletal muscle, as it's typically called, that's how we get that word, striated muscle, from striation, stripe. So what are these stripes? Well, the stripes are these myocytes that are fused together, so they're in series. So I'm going to represent the myocytes as these little squares. And if you take a look at an individual myocyte, I'm going to try to draw it here for you. It'll be from here to here. There's a dark part of the myocyte, and there's a lighter part of the myocyte. The dark part is called the A band, and the A band is formed of overlinked actin filaments and myosin filaments, the thin and thick filaments, respectively. The lighter part, and I'm here showing it for cell 1 and for cell 2, is called the I band. And in the I band, there aren't any of the myosin thick filaments. There are only actin thin filaments. That's what forms the striations, the stripes that we see under the microscope in muscle tissue. So each sarcomere is about 2.3 microns long, And when the muscles contract, those cells shorten. They become less wide by, you know, maybe 100 or 200 nanometers or something. But the combination of that in series creates a pretty dramatic change in the length of the muscle. And you can just experience this yourself. Straighten out your arm and get a feel for the length of your biceps. And now flex your arm and get a feel for the length of your biceps. It's much shorter when you do the flexion, and that's because of muscle contraction. Here's a better look at the striations and a better look at the A bands and I bands. Now we're looking at them under electron microscopy. And let's first begin up here, and then we'll relate it to the cartoons that you see below. So here we are looking at the A band, where we have the overlapping myosin and actin. And the part of the cartoon down here that is sort of the comparable part is the part that I've just outlined, where we have in green the myosin thick filaments and in red the actin thin filaments. Where they overlap, we have this electron density up here, and that's part of the A band. The M line, which I'm indicating here and here, is sort of the middle of the stripe, if you will, and that's where the thick filaments of myosin are anchored. Over here is the I band. So the I band for the first cell that we've drawn, and then the I band for the second cell is here. And you can see that they have less electron density. They look much more sort of white versus black. And that is because here there are no overlapping thick and thin filaments. There's only thin filaments. And here is what's known as the Z disc. If you're interested in this anatomy, I'm just giving you a quick little introduction. The Z disc is where the actin filaments get anchored. So it's kind of cool. So myosin is not a cytoskeletal component. It's not found in any other cell type. It's not found in, let's put it this way, it's not found in uncontracting cells. So any cells that contract will contain myosin thick filaments, but cells that don't contract will not. So it's not really a cytoskeletal component per se, but it's acting very much like a filament of the cytoskeleton in this example. All right, so what happens when the muscle contracts? Here is a contracted electron micrograph. So what you'll notice is that I band looks a lot thinner. Okay, look at the thickness of the I band here in the uncontracted state. Look at the thickness of the I band in the contracted state. And the A band is a little bit bigger. And the reason for that is now there is more overlap. And that's because the Z discs get pulled close to the M line. So the Z disc here, when the muscle contracts, is going to move in towards the M line. And the Z disc over here is going to move in towards the M line. And now you see the Z disc is closer together. And there's now more overlap of thick and thin filaments. That's because of the sliding filament action. Now the way this works, myosin filaments have a tail and then they have a head. And the tail is what the artist has tried to show in lighter green here in the cartoon. The head is what the artist has shown as the darker green part. That head is like a clamshell that opens and closes. And it can open and close and clamp onto a monomer of actin in the actin filament. And then when we're ready to contract, the myosin head changes its shape and it kinks about 90 degrees towards the M line. And the result of that is that shift in the shape pulls the thin filaments of actin closer to the M line. And we do it from both sides, so it's symmetrical, and it produces a shortening of the myocyte. All right, so how does this all work, and how is it all regulated? Now we're going to look and see kind of other proteins that help to regulate these dynamics. So what I mentioned in the previous slide is that the myosin head is sort of like a clamshell, and it can open and close, and it can grab onto a monomer of actin. But I want you to notice that sometimes the myosin binding site, which is shown as sort of the orange part of the actin monomer here, is unavailable for binding to myosin. So the myosin binding site on actin is covered up, and it's covered up by a protein called tropomyosin. We're looking at the filament sort of head on here. So if we could take the filament and then we could turn it to face us, this is what we would see. The tropomyosin is sort of covering up the actin, the myosin rather, binding site on the actin monomer. We got to move that tropomyosin out of the way. And the way that that works is calcium rises in the cytosol of the myocyte, and when calcium rises, it binds to a component of this complex, and then that pulls the tropomyosin out of the way and makes available the myosin head binding site on the actin monomer. Now that myosin head clamshell can grab onto the actin monomer. And then, as I mentioned, a conformational change in the myosin head can pull this in the opposite direction. So that's the sort of head-on view. This is the longitudinal view. Now we're looking at the actin filament. All these little red globular actins have been polymerized into a filament, and you can see them, you can see them sort of just lined up. They're very stable. They're in the ATP-bound state because of capping proteins that aren't shown here. The tropomyosin that covers up the myosin head binding sites is sort of spiraling around the actin filament. And it's covering up those myosin binding sites, preventing the myosin from engaging. The key regulator of whether tropomyosin is covering those sites up or not is a separate complex called troponin. Troponin's made up of 3 different subunits, the I, C, and T. The one that we care about is the C subunit because that one is the one that binds to calcium. So take a look here, kind of schematically. The myosin binding site on the actin filament is here, but it's covered up by tropomyosin. When we add calcium, notice how that moves out of the way, and now the myosin binding site is available. So calcium is what regulates that shifting dynamic. And if you're wondering how calcium binding to troponin C, right here, could pull tropomyosin out of the way, remember that all of these proteins are interacting through weak force attraction as kind of a large macromolecular complex. And since they're all touching each other, as the cartoon here is indicating, if we change the shape of one of them, we will change the shape of all of them. You know, if you start adding a divalent cation to the structure, you're going to destabilize the native or normal structure pretty dramatically. We get rearrangement of all the domains within these protein subunits, and that results in a pretty dramatic conformational or shape change. So if we add calcium to one of these subunits, it changes the shape of all of them. And the net result is to move the tropomyosin out of the way, allow the myosin head to clamp down on the actin, and now we can slide the filaments towards the M line. All right, that is the end of part 1. I'm going to take a short break and I'll be back for part 2. Part 2 will focus on microtubules and look at a specific example regarding APP. I'll see you soon.